Okay, perfect. Hi, everyone. I'm Svetlana Marukian. I'm Head of Discovery at Axcella Health, and I am very excited today to talk about an approach that we are taking in the drug discovery. First of all, I would like to thank the organizer and chairman for giving opportunity to share our work at Axcella with you all. So this is disclaimer. Just before going into the details of the presentation, I would like to tell you that Axcella's non-IND clinical trials, which are based on using the food study non-IND clinical studies, are called just, I'm calling moving forward as a clinical studies, referring to as a clinical studies. So let's start with the question that Axcella asked. Could we leverage endogenous metabolic modulators, which is metabolites and some of the amino acids and other metabolites, as a platform to actually harness complex metabolic diseases? It is well established that endogenous metabolic modulators and some amino acids are capable of changing clinically relevant endpoints in complex multifactorial diseases. And in fact, single and simple combinations of amino acids have been already used and demonstrated value in therapeutic settings. So what we are doing at Axcella, we are specifically targeting complex disease by leveraging the advances in system biology, machine learning, using combinatorial screening approaches with analytical tools to design specific combinations of amino acids. Tools that we are using based on published literature, scientific and medical, as well as proprietary primary human cell system in context of machine learning. So first, what we do, we understand the biology of diseases and pathways that are associated and changed in the diseases. And then we look which endogenous molecules could be changed in context of disease and target those pathways. Then we screen single EMMs and EMM combinations, which I already told that EMMs are endogenous molecules, to identify combinations and constituencies and in ratios that we could put together and have effect on complex diseases. For that, we are utilizing, to do those screens, we are utilizing primary human cell systems in conjunctions with metabolomic, transcriptomic, and secretomic analysis from those primary human cell systems. I will start with the preclinical data that was generated based on our platform related to NASH-NAFLD. So it is well established that disease progression is NASH and NAFLD is very complex and multifactorial. Insulin resistance, lipotoxicity are key elements driving disease, progression of disease. It is likely that multiple other pathways are involved in it too. Given the complexity of multifactorial pathogenesis of NAFLD, most likely a combinatorial approach is required to harness these diseases. So one way of utilizing safe combinatorial approach is using endogenous molecules that are safe. There are many examples of endogenous molecules like omega-3 fatty acid, various hormones, amino acids, their metabolites. And it is well established that amino acids are powerful signaling molecules. They are capable of regulating major pathways such as mTOR, AMPK, NF-kappa B, HIF1 alpha, and many others. Changes in amino acids are also directly linked to the type 2 diabetes and NAFLD pathogenesis. Amino acids have long history of safe use across multiple populations. And we are predicting that optimal combination of amino acids or endogenous metabolic modulators have potential to elucidate clinical results. So based on our platform, design platform, we to address dysregulated metabolism associated with the type 2 diabetes and NAFLD, we combined data mining, PK/PD modeling, and cell biology tools to design composition and ratio of endogenous modulators to affect pathogenesis. Through this process, we identify a combination of leucine, isoleucine, valine, arginine, glutamine, and N-acetylcysteine as the lead candidate in a clinic, as it is called AXA1125. So if you look at, this is our combination, and in clinic we call it as a AXA1125. We hypothesize that LIV-RQNAC can directly and simultaneously impact all three canonical pathways that related to NAFLD. So we test this hypothesis through multiple models. We have tested it in primary human cell systems. We did test them in multicellular systems based on primary human cells, in animal models, and we have also right now clinical studies. The results throughout all these models were very consistent. And I will start with the first model system that we use. It is hepatocyte model system. Primary human hepatocytes from healthy donors were used to drive to disease state by introducing saturated fatty acids and TNF. And you can see in this panel that when we treat with saturated fatty acids and TNF, there is a strong accumulation of lipids in the cell. Now, after we treat, in parallel of treatment with saturated fatty acid and TNF, if we treat with LIV-RQNAC, we can see strong changes in phenotype of lipid accumulation from microsteatosis to macrosteatosis. Not only we are changing lipid accumulation pattern, but if we look intracellularly at level of triglycerides, we are also reducing level of triglycerides significantly. And after the same system we used to test how AXA1125 or LIV-RQNAC, which is composition of the AXA1125, same lipotoxicity model in primary human hepatocytes to assess insulin sensitivity. Hepatocytes from healthy donors were similarly treated with saturated fatty acids and TNF, and then in presence or absence of AXA1125. We measured glucose output in the media, and as you can see, in presence of AXA1125, we have much stronger effect on the total glucose output compared to the vehicle. So AXA1125 or LIV-RQNAC having effect on glucose output. In addition, here we saw not only changes in glucose output, but also significantly increased phospho-AKT levels, and AKT being major protein in insulin signaling. Together, data from primary human hepatocyte indicate that LIV-RQNAC can simultaneously decrease lipotoxicity and improve insulin sensitivity, which are the core drivers of NAFLD progression. Next, let's turn our attention to inflammation, because as we said, NAFLD is multifactorial diseases and have multiple nodes, and one of the nodes is inflammation. To address how LIV-RQNAC could affect inflammation, we employed primary human macrophage-based system. Macrophages were driven from peripheral blood of healthy donors from CD14 positive monocytes and treated with LPS to differentiate it to M1 macrophage stage. And you can see here in the panel that LIV-RQNAC treatment reduces in dose-dependent manner levels of pro-inflammatory cytokine IL-6. And I will just here mention you that X here is plasma, 1X is plasma level of amino acids. These amino acids are present in plasma, so 10X is 10 times higher than in plasma, 20 correspondingly 20 times, and 30x is 30 times higher than normal human plasma levels. Media was adjusted to normal plasma levels of amino acids. And similarly, we can see effect not only on IL-6, but also we can see reduction in TNF-alpha production after LPS treatment in presence of LIV-RQ-NAC combination. Similar to M1 macrophages, in IL-4 treated M2 macrophages, which were driven from PBMCs as well, we can see dose-dependent suppression of CCL17. This is inflammatory cytokine that's produced by M2 macrophages. And conversely, it is inducing anti-inflammatory cytokine CCL18 in dose-dependent manner in 4 independent donors. So data from primary human cells suggest that LIV-RQ-NAC likely involved in metabolic reprogramming of macrophages from pro-inflammatory to anti-inflammatory phenotype. Next arm of the development of diseases is fibrosis. We also looked at the major cell that drives fibrosis in NAFLD-NASH. To induce fibrosis stage, we used primary human stellate cells from 3 healthy donors and treated them with TGF-beta. TGF-beta signaling cascades is directly linked to development of fibrogenesis in NASH. So in this slide, you can see that Axcella 1125 treatment or LIV-RQ-NAC treatment affected levels of ProC3, which is marker of fibrosis in clinic, dose dependently. It affected production also of ProC1, another marker, another collagen that produced by stellate cells. Not only it affected protein levels of 2 major proteins, but it also affected HSP47. HSP47 is the molecular chaperone required for collagen maturation, and it is active drug target right now. So next we ask question, okay, we are affecting fibrotic endpoints, and what could be potentially underlying mechanism of action, how it happens. In order to understand that, we used same cell system, and we looked at the activation marker, hepatic stellate cell activation marker alpha SMA. And you can see here 3 donors, and we can see dose-dependent reduction in stellate cell activation marker alpha SMA, as well as we looked at the EDU incorporation rate, which is LIV-RQ-NAC demonstrated dose-dependent reduction in EDU incorporation as well, which is proliferation marker. With this, we had examined all 3 nodes of the diseases-related signaling cascades in relevant cells. We also, next, we decided to test in multicellular system that consists of primary human hepatic stellate cells, macrophages, and hepatocytes. And you can see in the schematic how it was designed. It is pretty complex system, and in upper chamber, the transwell hepatocytes are plated, and in the bottom chamber, underneath of hepatocyte, macrophages and stellate cells. So we have well and transwell readouts. We read out from the upper portion as well as the bottom portion of the transwell, and assess effect of LIV-RQ-NAC at 30x concentrations from both chambers. And what we could see that LIV-RQ-NAC reduced secretion of inflammatory markers, IP10, MCP1, IL-6, IL-8, RO1 alpha in both compartments. And major effect on procollagen-1 was associated with hepatic stellate cells and macrophage marker, which absolutely makes sense. Data from dry culture were consistent across all monoculture data. Just give you a flavor how we design our combinations of amino acids. And one of the examples in this slide, now you would, I would provide you only one example in M2 macrophage context. In the left panel, you can see that here is our single amino acid, some of the singles And then from single amino acids, we are combining them together, make combination of 2 amino acids, more complex amino acids, LIV, RQ here, arginine, glutamine, DRR. And then also we have more complex, next level of complexity, LIV, RQ versus RQ-NAC complex. And then in the end, we came up with this combination of LIV, RQ-NAC. And I will explain you why here in this plot. LIV, RQ-NAC could be basically constructed by combining LIV with RQ-NAC. And you can see effect of LIV now on CCL18, which is inflammatory cytokine that produced by M2 macrophages, and RQ-NAC as well. So, and you can see that both of them are inducing this anti-inflammatory cytokine in the certain levels, but combination of LIV, RQ-NAC is higher. Now we can construct this LIV, RQ-NAC by combining NAC with LIV, RQ. You can see that NAC is inducing CCL18 to certain level, and LIV, RQ is inducing certain level. But when we combine those two together, we have much stronger induction of anti-inflammatory cytokine. So this is this example in one cell, but similar findings were observed using other phenotype readouts in different cell types as well. So with this, in summary of preclinical data, we demonstrated that LIV, RQ-NAC impact the multifactorial pathophysiology in NASH-NAFLD by direct modulation of metabolism, inflammation, and fibrosis. And it does so with consistency across all cell models that we have tested. Our platform allows us to deconvolute the diseases to individual cell type, to understand cell type specific effects of the treatments. We are also able to put these cells back together in organ specific ratios and architecture. Also, we are capable to deconvolute compositions to single amino acid and less complex compositions and identify best stoichiometry and ratio in compositions. Platform has already generated a number of successes in clinical settings, completed pilot 12-week single-arm clinical study in subjects with type 2 diabetes and NAFLD. We initiated a 16-week randomized placebo-controlled NAFLD study that readout of which is going to be presented soon. And also we are planning to go to pediatric setting because these molecules are safe to use and test our candidates in context of NASH, NAFLD in pediatric settings. So now I want to switch gears and give you a little bit of overview on some of the results from Axcella's clinical studies. So first, I would like to share data from pilot single-arm 12-week clinical study with Axcella 1125, which is LIV RQ-NAC composition. We used in subjects with type 2 diabetes and NAFLD. And here are results, just highlights from the results. Axcella 1125 administrations were safe and well tolerated. It is the administration reduced liver fat when measured by PDFF. Also suppresses inflammatory markers, ALT and MCP1, and we had effect on the PRO-C3 level, which is a marker of fibrosis. So together, this data indicate effect of Axcella 1125 in clinic on metabolism, inflammation, and fibrosis in subjects on over 12-week time period, exposed to over 12-week Axcella 1125. So next, next study that we are right now, it is the readouts of which is going to be in end of second quarter, is Axcella 1125-003 study, which is 16-week study, and it is 100 subjects over 100 subjects were randomized to Axcella 1125 and 2 doses of new composition, which is Axcella 1957, with placebo arm. So in this study, results of which is going to be presented in August at EASL, we identified that Axcella 1125 affects metabolism, inflammation, and fibrosis. With this, I just want to give you this in summary, the how Axcella 1125 works, or LIV-RQNAC works. Based on preclinical and clinical data, part of which preclinical data that I shared with you today, we demonstrated that Axcella 1125 impacts the multifactorial pathogenicity of NASH and FLD by direct modulation of metabolism, inflammation, and fibrosis. And LIV-RQNAC induces activation of cellular sensor, energy sensor AMPK, and you can see here. And this activation drives activation of PPR alpha, and in conjunction with amino acid catabolic pathways, and suppresses HIF1 alpha pathway. So global changes in these NASH-related major pathways, together with effects driven by individual amino acids of combination LIV-RQNAC, leads to improvement in insulin sensitivity, lipotoxicity, in conjunction with profound suppression of fibroinflammation. With this, I want to thank Axcella's team, that data which I presented today from Axcella, and also thank you everyone for your attention, and I am ready to answer your questions.
Thank you very much, Svetlana. Mm-hmm. If we have any questions from the audience, on a quicker note.
Yeah, I have a question.
Yes.
So how I have a question about the PK of the combo, right? So in your preclinical studies, these are all cell-based assays. And then in your clinical studies, how do you make sure that the combo has the same exposures as in your cell study? In terms of each individual amino acids, they have the same ratio as in your in vitro studies, since they have different pharmacokinetic properties and in the different tissue distribution. How do you guys control that, and how do you guys check the pharmacokinetics in the clinical studies?
Yeah, that's a very good question. So first of all, I didn't show you all the data that we have. So we have done PK studies, rat PK studies, as well as in human, we have some PK data. So we looked at the plasma exposure, and what we can say that plasma exposure in rat, the doses that we are using is close from lower doses 10, close to the peripheral blood, and higher doses 30, it is more a portal vein exposure. So the range that we choose of exposures was based on what is in preclinical rat model in the portal vein versus in periphery. So we definitely look into that.
So how do you deliver your treatment?
That it is sachet. Basically, it is we have the dry mix that needed to be water is added, and drink as a basically drink as any other drink.
Okay, thank you.
So this is Zoe. Do you classify this as nutritional supplement or medicine?
Right now, we declare it as a medicine. We are going as a medicine since basically initial studies were done as a food studies, and that's why we called it a clinical study, not clinical trial. And we are moving forward with it. Already declared as a medicine.
Thank you very much.